the human genome and this new field of bioinformatics. So he was thinking, God, this might be a good place to land. Good possibility, you know, listening to this NPR news, our news broadcast, and started to look around on the internet and found out there was only a couple places around that offered anything. Most of them were biophysics and things like that. And there was one place in Michigan that had something called bioinformatics, and he thought that was where the future was. And came here. He was absolutely right. And, you know, his very impressive CV, I think, with over 200 publications, proves that he was right. And he's established himself as a leader in cancer bioinformatics. He's made numerous contributions to the TCGA project and has led several analyses, you know, in tissue-specific and disease-specific areas within TCGA. He's participated in several dream competitions over the years, you know, and he's been involved in numerous collaborative grant and contract proposal efforts. It's really impressive what you've been able to do there. You've established yourself as a bioinformatics analyst leader, collaborative leader in cancer bioinformatics, where you now serve, you know, at Baylor College of Medicine as an associate professor. And we're very proud that you're our graduate. We're very happy that you're back, and we're delighted that you're interested in our new faculty director recruitment, you know, for the bioinformatics core, working with the fine team that Rich and others have established over the years, and with Cassie's leadership. So welcome, and we're delighted to hear about the Cancer Genome Atlas, a resource for molecular biology studies. And, you know, it's great that we have an expert to tell us about that, because it's so important to our thinking about cancer bioinformatics in our future, thinking about bioinformatics services and bioinformatics research in cancer and beyond. So welcome back, Chad.
Yeah, thanks a lot, Brian, for that great introduction. It's always, I came back here in 2012 to Ann Arbor to give a talk, and it's been about 4 years, but it's always nice to come back, and it's been a real treat meeting a lot of familiar faces. Been a lot of changes on the campus over the last 10 years. All of it seems good. You know, just a lot of growth and a lot of new possibilities. So it's very exciting. Glad to be here. So for the science talk, I'm going to go over the Cancer Genome Atlas and kind of focusing on my own efforts within this large consortium, this very big science project. Kind of focusing on, you know, thinking about, you know, molecular biology or understanding of the molecular biology of cancer as it relates to these big genomic data resources like the Cancer Genome Atlas. And I think there's a, you know, a couple of themes in this talk, and you know, going through different results and different different studies. But you know, this idea that these big genomic data sets, like from TCGA, have you know, we can get different insights from them based on our domain expertise and our knowledge of you know, molecular biology of cancer and the pathways that have been established over the last few decades, and that's a knowledge resource that we can apply to these cancer data sets to gain new insight. But even, and then the second part is, you know, after, you know, in the wake of these big data sets have been generated and the initial study has been completed, the data are in the public domain, and they'll continue to be used by my group and others in you know, in ongoing molecular biology studies, maybe studies that aren't necessarily built around a big cancer genomics data set, but have very make extensive use of experimental models, and how can we leverage these resources from human tissue to our understanding of these experimental model systems, which can kind of further extend our knowledge of the molecular biology of cancer. So as an overview, the Cancer Genome Atlas, or TCGA, is this large-scale collaborative effort to systematically characterize the genomic changes that occur in cancer. And it's carried out now comprehensive molecular profiling for over 10,000 human cancers. Not just sequencing, but there's several molecular platforms all carried out on the same set of samples. So lots of opportunities for data integration. These data are all available now, and there's no restrictions on publication. We have data now for, I think, 32 major cancer types, representing, I think, over 10,000 samples of data are freely available for any platform, posted on a couple websites where researchers can get level 3 data that's ready to analyze and plug into with new questions in mind. So a lot of the effort in the last several years from TCGA, not just generating the data sets, they would have some kind of publication moratorium on the data, and then maybe spend 1 or 2 years putting together sort of a high-level publication featuring the data set on a specific cancer type, and that could go into Nature or Cell. And this is some examples here listed. I think each cancer type got its own publication initially. And I think these studies, I think they gained some new insight, but in a lot of cases, they're just really scratching the surface, in terms of a lot of the questions we could be asking. The way these, TCGA, projects are set up in terms of the analysis that goes into the major paper, marker paper, is, this interinstitutional collaboration, and oftentimes via teleconference calls and a shared wiki page, where analysts can upload results, and week by week via call from maybe 30 different institutions. It could be up to 300 or more authors on a TCGA paper. Not all of those necessarily be involved in the downstream analysis, but I think you could have what we call an analysis working group of maybe 30 or so investigators that meet on a regular basis and share results, and then think about what's of great importance to highlight in the resulting paper. So I'm going to focus on the next several slides of examples of some of my own contributions to the TCGA marker paper. So these are big papers with lots of authors. I didn't do all the work, but we would try to find maybe an opening where maybe we had some an angle or some insight that we could contribute that I guess was important enough to highlight in the paper. So kind of building on just my own work, which doesn't cover everything that's done as part of TCGA. So kind of my first sort of cut my teeth as part of TCGA with the ovarian cancer paper, which is the second paper. I missed the glioblastoma paper, but then I was involved with other collaborators in ovarian cancer, and the Genome Center saw what I was doing and wanted to pull me into getting involved with TCGA. And initially, just the ovarian cancer project was the second project they TCGA undertook. So my contribution to that paper was, I think, very maybe a straightforward training testing scenario, and trying to define a gene expression signature of prognosis or patient survival in ovarian cancer. So you can take a training data. Take a training data set. That's what I want. So the training data set, and then define genes positive correlated, genes negative correlated with outcome. Apply that to a test set, and kind of see if that is able to predict outcome, and basically just using methods I was taught here at Michigan. And not only can we plug that into TCGA data in a test set, show that reliably predicts worse versus better prognosis, but even there's other public data sets of ovarian cancer, so we could take those and take the same signature and kind of show how it's consistently reproducible in other data sets. Now that sort of represents information. There's raw information in terms of a set of genes that are coordinately expressed in maybe more aggressive cancers. But what would that tell us? In terms of the pathways of the processes, that's sort of a higher level, kind of a next step for this type of analysis. What does it mean in terms of the pathways? And so, as we move on to think about kind of this bigger question of of pathways in cancer using genomics data, we have to ask just how do we really understand how molecular pathways work in cancer, right? I can think of there being two kind of major sources of information on pathways. a lot of work in terms of understanding these pathways in cancer have been built up using experimental models. It could be cell lines, it could be mouse models. These could be perturbed in real time in the lab to establish cause and effect relationships. But then the caveat always with experimental models is that the relevance of the human disease is not always clear. That can be a critique that can come, if one wants to play, be a bit critical. If data is just coming from a mouse, how do we know that that relates to cancer as it manifests in a human patient? So we can get data from human tissues, like from TCGA, and we can look at correlations and establish correlative relationships, so not necessarily cause and effect, where the relevance to human disease is arguably more clear. And so a lot of my work and a lot of my contributions to papers is really trying to bridge these two sources of information. So you can kind of leverage the best of both, and any high-level publication is going to have elements of both. You want to establish, maybe what you think are cause and effects, and also show that there's something in actual human specimens that this is consistent with what the experimental models would predict. So, for example, here's a cartoon of the PI3 kinase pathway. This is maybe one representation of the pathway, and this would be maybe the accumulation of knowledge kind of built up over many research articles and research labs, and kind of what's the current model over the years. And the question is, well, how does this relate to cancer? Are all of these nodes in this pathway relevant in cancer? Are they all being equally targeted in terms of the genomic alterations? How does it translate in terms of the transcriptional alterations or the protein signaling? How does the pathway look maybe at these different levels of data that we can explore using, say, TCGA data sets? So we made some inroads in this, and we're still working in particular on PI3 kinase pathway. But some of the previous work, first step into this was looking at breast cancers. This is the breast cancer marker paper. I had a couple figures in this paper. This is basically a pathway-centric view of PI3 kinase pathway-centric view within breast cancer, pulling out relevant genomic alterations such as PTEN loss or PIK3CA mutation. So these are DNA alterations. And then another level of data looking at the protein data with RPPA, which is a really nice platform. It's got a set of phospho antibodies for the representing the PI3 kinase pathway substrate. So that's how the pathway looks like at the protein level. And some of these are suppressors here at the bottom, and then we have some activators here. So there's a lot of red and versus coordinate blue on this side. And then we can look at gene signatures and how it looks in terms of how it affects the transcriptional pathway. So we can pick out subsets of breast tumors that align well with kind of the canonically accepted molecular subtypes, say the basal-like subtypes seem to be the strongest in terms of the pathway manifestation. But we're seeing here that maybe the genomic data doesn't necessarily completely explain what we can see at the protein data. Maybe a PIK3CA mutation doesn't always correlate with greater activation. And so this is where the integrative approach is something good to apply to, say, studying a specific pathway in a given cancer type. So we've extended on this, and we've kind of taken it all the way to looking at all the cancers in TCGA. This is a pretty ambitious project. This is probably TCGA's last major endeavor, called the PanCan Atlas project, looking at all 10,000 human tumors. They've been all kind of systematically analyzed on the same data platforms, and now we want to put things together. It's too big of a data set for one paper, so we've scoped out a PI3 kinase AKT mTOR paper where we're not just starting with the data, we're starting with a model of how we understand the pathway operates, and we've got David Kwiatkowski and others who are respected molecular biologists. They understand how kind of the mechanics of the pathway, and Gordon Mills has just awesome in terms of the proteomic data sets that he's generated. And then my group's kind of putting it together. But here we have basically some of the major members of the PI3 kinase pathway in terms of the alterations and how they are typically altered. in significant subsets of cancer in terms of, maybe the mutation versus the amplification versus, copy alteration. And, this is not just limited to one gene. It's several genes that maybe, can explain an additional subset of cases that would appear to be altered, at least in one gene for the pathway. And how do we know that we're getting this right, that we've scoped out this model? Does it really explain, is it really reflective of the pathway as it truly is and as it's truly manifested in cancer? Well, I think this is where the integration can help us. And this panel here at the bottom is RPPA data, the proteomic data. In theory, if this pathway is more active with these, say, these genomic alterations, it ought to show up somehow in general, and broadly speaking, in the proteomic data. So we've got several key protein features that reflect the pathway, and mTOR's signaling is kind of along the bottom. It's a little more red here, you can see by eye, and more blue on this side. Cases that don't have anything. There's a couple surprises of this analysis. Maybe like single copy losses of P10 or STK11 look to be important in terms of the correlations that we see. So it's not just mutation, but I think we need to consider other modalities. But there's also a subset of cancers that have very strong activation of the pathway at the protein level, and it wasn't there wasn't a canonical genomic alteration that we could use to explain that. So that means that some of the other things that may be acting on this pathway, and in fact, kind of doing a deep dive and some data mining, we picked out a couple genes, one of which was VHL, which was nobody believed, and then last week there's an article in Cell showing that VHL directly interacts with AKT. So, we found a hypothesis, and then it was like proven by somebody else almost simultaneously. So this is where there's some discovery that can go on here. But in general, the pathway, as we understand it, is it fits well when we put all these data together, and I think the model, as we understand it, is holding up quite nicely by and large. So the next set of analyses we did, I did a lot of work in kidney cancer, and in particular, in the kidney cancer, they divided up into three major different cancer types, so each type got its own paper, and actually they're very different in terms of biology and their genomic profile. But I wanted to extend the analysis that we did in the ovarian cancer and maybe dig a little bit deeper in terms of understanding things in terms of pathways. And this is where we apply to clear cell renal cell carcinoma, or CCRCC. So here, we took the analysis in the ovarian, but also extended it to other platforms, looking at mRNA, microRNA, DNA methylation, and protein by RPPA. So each of these platforms we could separate into discovery and validation cohorts, and define correlates of survival and for each platform, and apply to the validation data set. Yeah. Okay, I'm sorry. That's so I showed that in the previous slide here. So it's like the proteomic data. It's nice because it's only like 200 protein features, but they're targeted for the ones that are of particular interest in cancer. They're very highly enriched in, for instance, in PI3 kinase pathway members. But you get the phospho levels, the activation levels of the protein. You won't see that in mRNA. It doesn't really correlate with mRNA. So that's another level that we can understand. The pathway, not just if it's expressed, but if it's activated. Thanks. So for each of these platforms, we could reliably distinguish higher risk from lower risk patients, or more aggressive from less aggressive kidney cancers. And this game has been played a lot in terms of just trying to find a molecular signature of survival, and if you improve on the the clinical standard clinical models in a way that would be enough to actually affect a change in treatment. That's a pretty high bar. It's not really why we did this. I think we thought, well, there's information here in terms of what are these genes, what are these gene features, what do they tell us, and how do we put this together to kind of get some deeper insight. So that took that took some thought, and I think we kind of got our start kind of digging through the proteomic data and found that there's a lot of metabolomic, metabolic related features in there. In fact, the protein survival correlates included AMP activated kinase, or AMPK, and acetyl-CoA carboxylase. They're sort of anti-correlate with each other. One, loss of one is associated with more aggressive, and then overexpression of the others associated with more aggressive. But these interact in an important way. You know, this low AMPK combined with high acetylcholine carboxylase here is then associated with worse outcome. You know, downregulation of the one leads to upregulation of the other, which can lead to a metabolic shift from oxidative phosphorylation to aerobic glycolysis. And in terms of our understanding of pathways, that can represent a type of Warburg effect, which is a phenomenon that's been talked about for over 50 years in cancer. So we thought we were onto something here, and we got together with other investigators in the group who knew the molecular biology really well and kind of gave them a start, and then they gave us some pointers, and a very collaborative relationship to try to piece together the entire pathway and looking at it in the context of metabolism. So these core metabolic pathways that we've well known and well studied, but on this we've overlaid directly data from TCGA where, say, a red node would indicate a correlation of a gene or protein with worse outcome, blue node correlation with better outcome, and you have protein or RNA or DNA methylation features. When we put everything together, we found that these survival correlates do indeed underlie a type of glycolytic shift. So the direction of the changes is really important. So, for example, we have upregulation of genes related to the pentose phosphate pathway being associated with worse outcome. And then we have downregulation of AMPK and different components combined with upregulation of genes involved in fatty acid synthesis, also being associated with a worse outcome. And then in the mitochondria, where we have oxidative phosphorylation, there's a systematic downregulation of oxidative phosphorylation genes associated with a worse outcome. So this is kind of a prime example for me in how we can move from raw, kind of a catalog of molecular correlates, which the TCGA is primary motive in trying to give the community, but then trying to move from those molecular correlates to some true pathway level insights. And and you can get that if we put these in the context of our understanding of molecular biology and find a good context to put these data into. So moving on, we did another project in chromophobe kidney cancer, and then we asked a different question here, and this is also involved integration, but a different type, to understand the cell of origin of kidney cancer. And this is a project I co-led with Kim Rathmel as part of TCGA, had a lot of support from the Genome Center at Baylor, but also with other investigators at other institutions, trying to understand the somatic genomic landscape of chromophobe renal cell carcinoma, which is more rare as compared to the clear cell type. It's got much better outcome, and actually, there isn't a lot of genes mutated. It was actually kind of challenging to kind of tease out some interesting stories, but we were able to make it happen. And one of the interesting stories was that we can compare these two types of kidney cancer. We've got clear cell renal cell cancers, or CCRCC here. We've got CHRCC here, and compare them, say, by DNA methylation or gene expression. It's the same story. Widespread differences, maybe involving like tens of thousands of genomic loci in terms of the DNA methylation landscape. And that kind of raises the follow-up question: is like, why are these cancers so different? So one idea would be like, well, there's different cancer pathways that are genomically altered in each type, and that's what's showing up in the profile. And that could be, but I think also maybe, especially when we think about DNA methylation, maybe this could be reflective of the cell of origin of the kidney cancer. Maybe this has a different cell of origin than this one. But how do we get at what's the cell of origin on the basis of molecular profile? Yeah. Huh? I think for the DNA methylation arrays, it's not. They're just genomic loci, so they may be associated with a gene, but they're just different spots in the genome. Some of them are within CPG islands, which are canonically associated with epigenetic silencing. But they could be all over. They're not necessarily tied to a specific gene. They're just kind of all throughout, and they're not necessarily limited to one location. You can. Not all of these necessarily translate to the gene expression data set, but many of them would. Yeah. Yes. Mm-hmm. So and there's this notion in the kidney cancer community in terms of the cell of origin of kidney cancer, and there's a hypothesis out there whereby they arise from different cells within the kidney nephron. So here's a diagram of the kidney nephron. It's just part of the kidney, but it's part that processes urine from the blood. And we can just think of this talk. There's a proximal end on this end, the kind of snake-like end, and then a distal end here on the right. And the theory that had been put out and debated is that the clear cell kidney cancers arise from specific cell types within this end of the nephron, the proximal end, where the chromophobe arise from a very different cell type at this end. And this has been, there are some studies using immunohistochemistry of specific cell markers, and that was sort of an explanation for those data, but not everyone was satisfied with that, with the current data, and we wanted to maybe use these data sets to ask to do a global analysis to try to get at this question. And this is where using data in the public domain and kind of this notion of people making the data sets publicly available was a great boon for us, because someone a group had gone through the efforts of deriving this gene expression atlas of the kidney nephron. They'd taken kidney sections from mouse and human, micro-dissected them, and profiled them for gene expression, published a paper, made the data available. And so this is something that we can have done ourselves, but it was already done for us. And we wanted to take this data set and look at it in the context of our kidney cancer data. And so when we did it, we got an interesting result, a very interesting result. So we have here, we cross the nephron data set with our kidney cancer data set. We find that CHRCC versus CCRCC expression differences reflect distal differences between distal versus proximal nephron, respectively. So here, we've taken here on the rows represent the kidney nephron sections. So we have proximal sections and distal sections. And then the columns represent our TCGA kidney cancers. We've got maybe 500 samples here. We've got maybe 450 clear cells here. We got like our 50 or 60 chromophobes here. And we do a correlation between each differential profile from the nephron and each differential profile from the kidney cancer. Red denotes a kind of a global similarity. Blue represents a dissimilarity or anti-correlation. And I think, do this for each individual sample against each individual nephron section. And the results are very clear, very strong, that there's strong correlations of the clear cell cancers with the proximal sections of the nephron. So a lot of similarities between what's expressed in the proximal versus distal versus what's expressed in clear cell versus chromophobe. And then the chromophobes show up as very distinct in the distal section. And it's probably a clear result as we can get. But it also is very consistent with what many kidney cancer biologists would understand. And I think in terms of the debate, I think it actually swayed the debate more towards the side of different sections giving rise to different kidney cancers. So there actually was 3 kidney cancer subtypes. They're all very different. The way the TCGA was structured is like we did one paper on clear cell, one paper on chromophobe, one paper on another subtype called papillary. So I had heavy involvement in both in terms of either co-chairing or leading the analyses. But then it's like, well, now we've got all the data. Let's put everything together. What do we learn if we put everything together? That's not something TCGA was necessarily planning to do, so we just wanted to do it ourselves because we wanted to see what we find. And so this is a recent paper that came out earlier this year where we take maybe close to 900 kidney cancers from the 3 sub-projects, and we put it together and just ask, well, how we understand these are different in terms of the histology, but what can we learn when we put it together in terms of the molecular landscape? And I'm not going to go through everything that we did, but we did basically use all 5 of the platforms used for molecular subtyping, and then put the calls from each platform together in this kind of method called COCA, or a cluster of clusters analysis. And we could pick out specific subtypes. This is putting everything together, and we've got like close to 900 samples here, and then different platforms here, a different color represents a different platform, and a platform may make a subtype call. But it's with different platforms, it's like, well, what's you can look at the trees, but then you want to look at the forest and get the big picture. So we kind of put everything together in this way, and we come up these 3 histological subtypes. We come up with 10 major molecular subtypes. Clear cell is distinguished from chromophobe, is distinguished from papillary on the right. But then within each major subtype, you can break it down in terms of differences within each histological type that doesn't necessarily show up by the histology view. You can pull out expression features that kind of distinguish these groups, and maybe there's information here in terms of biology. Look at the DNA methylation landscape. There are some that show a very hypermethylated phenotype, a fair subset of the papillaries, and a lot of the more aggressive, what we call more aggressive subtype of clear cell kidney cancer. And then you can kind of map, look at things in terms of specific features, or cell cycle pathways, or EMT and VHL. A lot of individual genes that would show differences as expected. And then this is figure 1, figure 2 of the paper, and then the rest of the paper is really fleshing out what's the biology or the pathways unique to each subtype. I'm just going to go into one aspect that we looked at in terms of gene signatures of immune checkpoint pathways, and we explored that in collaboration with some people at Memorial Sloan Kettering, Ari Hakimi and James Say and others. And they had a great idea, and they'd already they kind of been exploring on their own, but they as part of their own studies, but then they very generous in attributing their results to our effort. And this is again using public data sets where a group had just profiled normal immune cell types, and there's a wide variety of immune cell type populations. Each one has its own distinct gene expression signature, and so each you can kind of come up with a signature for each immune cell population. Well, just like we did with the nephron data set, we can take this and maybe overlap it with the kidney cancer data sets. When we do that, I think it's this is getting at not just patterns in the tumor itself, but also patterns that may be representative of immune cell infiltrates. And the differences, when you saw a lot of stark differences here at the top, we have some immune checkpoint genes that are very standard, most commonly discussed or would be often considered as targets or for immunotherapy. Kind of individual genes here at the top. Some of these would be expressed in the tumor cells, some presumably expressed in the immune cells. And then here we have our immune cell type signatures. I mean, there's some differences in different immune cell types, but most of these showing up very hot for the subset of clear cell kidney cancers as compared to the other subtypes. Also, we can kind of see it in the methylation data. We're able to kind of tease out immune cell signatures in the DNA methylation as well, so it's consistent. And I think it's so we can kind of break it down and show it in terms of the pathway view, which is helpful for people. And we can look at individual markers like PD-1 or PD-L1, and clearly there are differences within kidney cancer subtypes. Maybe very high in the clear cell, but even within clear cell, there's distinct differences. Maybe the more aggressive, the red CC3 subtype is maybe shows even a greater manifestation of the immune checkpoint pathway as related to other clear cell or other kidney cancers. So in terms of where these genes presumably fall in terms of the pathway, some of these would be coming from the tumor cells. It turns on ligands to send signals to the T cells not to attack the tumor cells. So there's some coordinate activity where the tumor cells are turning on specific genes, and then the T cells are presumably very active, and they also have these expressed, and because these are coordinately on, and then they interact, and you have an invasion of the immune system. Individual markers delineate more aggressive from less aggressive kidney cancer. So it's not just metabolism, but you also see, like, put in the context of another pathway, then we can understand that the immune checkpoint pathway is also something that's being at play within more aggressive kidney cancers. Now, we've recently done this again in lung cancer. I guess this paper is in press, but we got warmed up on kidney cancer, and we had interest in lung cancer. We had collaborators in lung cancer, so it's like, well, let's just, TCJ is not going to do it, so let's just do it ourselves. So, no, I think, these data sets, that started out seeming incredibly big, but I think the more we work with them, the more others work with them, they feel more manageable, more tractable. And so this is, I think, pretty much all the analysis in this paper was my group, including pulling together the different platforms and, again, calling subtypes. Here we get like 9 lung cancer subtypes that span both the adenocarcinoma and the squamous cancers. And, we pull out gene features and spend the rest of the study kind of understanding in terms of pathways. I think some things came out of interest. We got a subtype that was mixed between both adeno and squamous, also poorly differentiated, or associated with poor differentiation and poor patient survival. Some of these subtypes are more associated with non-smokers versus smokers. Interestingly, and MAP kinase and PI3 kinase pathway were more active in some subsets versus others. I think for each of these subtypes, we could, there would be some therapeutic implications in terms of, in theory, there's something out there. Is it, immunotherapy? Is it a MAP kinase or PI3 kinase that would be, or even standard chemotherapy that might be brought to bear on that? That subtype might respond better to that therapeutic approach versus some other subtype that's expressing another pathway. So this last slide is something I want to get into in terms of the future and in terms of where, something we've been using PCJ data for and will continue to do so in the next, I guess, in the foreseeable future. This is not a genomic study. This is a paper that I was participating with my collaborator, Don Gibbons at MD Anderson. It was published earlier this year. It's basically a molecular biology study. I mean, it's nice work in terms of looking at molecular models. These collaborators have great models, mouse models, and they've done all the omics on them, and we've kind of spent like the last 10 years kind of going over it with them. It's been a nice endeavor. But this is where he had the paper mapped out, but what was missing was the human tumor component. So yeah, there's this pathway where there's this gene called Kirkle. It's a miR-200 target, and it regulates endocrine-dependent signaling. And so the mechanics and the functional relationships were kind of established in the lab, but he needed another piece. He needed to know, well, okay, I'm saying this is associated with EMT. It's a kind of maybe a new player in EMT and miR-200 targeting. Can we look at the human tumor data sets and see if that's really reflective of what we're seeing, reflective of the human tumor studies? So the panels in this one figure, 3 of these would be from my group, and then the rest is really kind of putting the complete story in terms of the functional aspects. And so that's kind of the experimental models, and then we have the human tumor data. You look at correlations. Okay, there's a subset that are consistently correlate with EMT markers and predicted miR-200 targeting. You actually get a narrow set of lists if you, depending on how many filters you add. Kirkle is one of those genes, interestingly, and you're looking across maybe close to 9,000 human cancers here. That you look at Kirkle versus expression of EMT or miR-200, that the correlation patterns are quite consistent. Looking across all human tumors, those that have expression of Kirkle were associated with more aggressive cancers, not just limited to lung cancer, but looking at other cancer types as well. So these here, we're making use of the TCJ PanCan Atlas, the complete TCJ data set, and we can ask questions that wouldn't just be limited to lung cancer or any one cancer type, but say, is there correspondence within like other tissue types and not just one cancer? And the TCJ gives one a quick way to kind of survey across all the different different cancer types. And we continually get asked questions about what, if someone has a gene, what they're doing in TCGA. Having done all the work up front and getting familiar with these data sets, it's not a lot of more effort for us just to pull out for a given gene. And so I guess until other people figure it out and they start doing what we're doing, then we'll just keep doing this and get out other papers. But I think it's going to continue to be a resource, and I think it's going to be a long time before we get a data set like this. So I think there's still more to be made out of these data. So that brings us, I think, to the end. I'll acknowledge some of the funding sources that specifically allow me to do TCGA research. So it's been a nice opportunity, good set of collaborators. And I'll give a personal update, those who knew me from when I was back here. We're up to 5 kids now. I think that'll do it for us. But it's been a lot of fun. So people ask me, well, what are your outside interests outside of work? I used to have outside interests, and now it's all these kiddos. But it's great. So I thank you a lot for your attention. I'm happy to answer any questions. Yeah, Dr. Ovarian.
Do you download all the data sets for the studies you intend to analyze onto your own systems, or do you do some of the computational, especially the preliminary analyses, in the TCGA site?
So the way TCGA is set up, there's level 1 data, which is kind of the raw data, and level 3 is kind of the analysis-ready data. We primarily work with the level 3 data, and then we kind of start with that to do pathway analysis. It's nice that I think the individual data centers they're required to get things to level 3. The full data sets are available, and so groups could go and want to do analysis on level 2 if they're trying to extract specific mutation information, say from the BAM files, in maybe a more specialized or more specific analysis, and that's doable for some of the data types. One needs to get permission, if it's going to be patient-controlled data. But I think for a lot of the work that I showed, I mean, you can start from level 3 data, and it can go on a desktop, a high-powered desktop, and because it's all like in tables. And I think for the 10,000 cancers, we have to kind of work a little bit differently. People don't really notice when they see the results, but I think it's all tractable because it's all in tables and putting tables together.
So are you also using the newer Cancer Genome Commons? Do I have the title right?
They replaced the data portal with the data commons, and so we've started to get into that. I think it's getting worked out. I think that's just a way to download the data, and we kind of figured out how to get that. But I think the Broad Institute, one of the websites I put up there early on, the gdac.broadinstitute.org, that's got a nice portal to the data, and we use that a lot because we just want a table of the data. And I think that the commons is a little complicated because it's like, I'll just give you one file at a time. And it's like, can you just give me a table? But yeah, we can work it out. It's just a learning curve. Yeah.
Anybody else? Chad, let me ask you about future direction. What do you see as major challenges for cancer genomics that you will have a chance to address, say, next 5 or 10 years?
I think, in terms of, where the data sets need to go, I think the TCGA approach could be applied to, individual studies. I think when it's big like this, like everybody's working on it, but I think there's a lot to be made just from individual studies. And what will make those distinguishable, I think, from Cancer Genome Atlas is, the clinical annotation of these samples isn't always great. Or, maybe the follow-up is like 1 to 2 years. So it's hard to ask questions. You can ask questions of what's mutated across all cancers. But, I think there's interesting questions in terms of, looking at clinical trial samples, which ones responded to therapies, which ones did not, what's the molecular pattern look like after therapy versus before. And some of that could be applied with the multi-omics approach, but I think, with the clinical data sets of better clinical annotation, there's maybe new questions that could be asked that we really can't ask. Oh.
I'm kind of curious about the relationship between ICGC and TCGA. My impression is that the ICGC projects has a bit longer trajectory. And do you think that there's going to be additional cancer-specific data sets coming down and maybe integrated with TCGA over the next few years, or is that going to kind of stop next year like TCGA will as well?
So the ICGC is kind of following its own path, and they've done a lot in terms of an international consortium, and maybe they get some of the same cancers that TCGA does, but they might do some different types. But there's already working together between TCGA and ICGC. I think TCGA, those letters are going to be retired, I think, this year or early next year, but I think it's still, mainly people like me who are involved with TCGA are involved in ICGC, and they're doing now an analysis of whole genomes, which we really didn't get into here, but a pan-cancer analysis of whole genomes, looking at like 2,800 or so whole genomes, and I'm involved in that. We're sort of got a study that we've scoped out that looking at structural variants and how they relate to gene expression. So that's and that's been going on for a few years now, a couple of years at least, and I think we want to get the papers out end of this year. So hopefully that will be like another nice big data set where it's not just TCGA. They just looked at the whole genome, so that's maybe like 1,000 from TCGA, like I don't know, 1,800 from other other countries. Kind of put together, get a common data set. They actually realigned all the RNA sequencing to kind of get a common data set. So that's been nice because I think the whole genome kind of lets us ask different questions, and that's another kind of big science, team science initiative where the data sets are kind of made available to everybody, and then I think and then people kind of can deep dive into it with specific questions in mind, and then publish a big paper, but also publish a lot of satellite papers sort of at the same time. Oh, at MD Anderson or just? Yeah, I'm excited for it in terms of the data sets, and I think MD Anderson got a lot of, well, I guess they got a lot of criticism, some criticism from some circles early on, but I think just in terms of where we are in terms of the science, I think it's a fantastic opportunity. Some people will ask, well, why did we do TCGA and what do we learn from it? I mean, I think it's continuing to be a science resource, and there's definitely, the data set can just only get bigger and we can only learn more as they get bigger. I mean, I think it used to be, when I was a student, like a data set of like 70 lung tumors was like massive. And now it's like, 10,000. We're starting to just get comfortable with working with 10,000 tumors. We have something with better clinical annotation and can really drive home the precision medicine. I don't know necessarily we have all the data to really like, make, firm conclusions. And so I think it's an opportunity that we just can't let pass us by.
So, Chad, when we ask you about what we should be doing next, what you're going to do next, a few other things come to mind. First of all, in the old days, gene expression meant microarrays. Now there's massive data with RNA deep sequencing, from which it's quite feasible to analyze splice isoforms and other variants, especially splice isoforms, which can be tissue specific, can be cell type specific, can be organ specific for certain cancers. And I think there's a lot more probability of informative biomarkers and highly targeted therapies at the isoform level than at the presumed gene mixture of isoforms that's the standard RNA expression approach. And of course, then there's the matter of intra-tumor heterogeneity. There's a lot of people working on that. And that really is not just a matter of description, that requires a strategy, because it's not simple statistically, and it's not simple biologically. And it's very time dependent and progressive. How do you think about those two topics, splice isoforms and intratumoral heterogeneity?
Yeah, I think it's all definitely within the realm of possibility, and I think probably, I think and so that's sort of another avenue that maybe TCGA doesn't cover. I guess it can in terms of the splice isoforms, but then it's like, what do we do with these in terms of how do we validate these? And an investigator that has an experimental model, they do RNA-seq, maybe they want the gene expression, and then they ask for the splice isoforms. But to work up the splice isoforms in terms of biology, like this splicing is different, because this has this function, and it takes a lot of work in terms of microbiology lab, and I think it just takes maybe some biologists, a willingness to kind of invest that time. If we have collaborators that are willing to invest that time, we'll meet them halfway.
Your second question is about intratumor heterogeneity.
Mm-hmm.
On that.
Oh yeah, I think well, it's so we definitely can learn a lot from doing different subsections of a given tumor specimen and look at them genomically and maybe understanding how one part of the tumor may be different from another part of the tumor. Even doing like single cell sequencing and trying to trying to get an idea of the clonal landscape of the tumor. Where maybe you're treating the most of the cancer with a given therapy, but there's like 5% of the cells that have something else, and they're going to dominate. And if there's some way we can predict that, so I think that's going to be something definitely in the future. I can't do it for everything, but I think at TCJ we still learn. I mean, some heavily criticize TCJ for not doing that, but I think we do learn a lot in kind of having a lot of samples, and you get, I guess you get a sense of the diversity maybe you would find within even a single tumor specimen. Some of that diversity we see across, like say, like 500 kidney cancers, you can find some of that even within a single kidney cancer sample. Some of it may have a more aggressive molecular signature than some may have a less aggressive within the same tumor, and that's something, I think in TCG we kind of get an idea of the landscape of all the different subsets, and some of those could exist within a single tumor, but at least we can get the idea of the molecular landscape of the TCGA data.
Questions do you have, or do biology-oriented collaborators come to you for help to address that we really don't seem to have enough analytical power or ideas to answer? What are the hard questions, the NP questions?
Well, I think a lot of it is we work a lot with investigators that really have a good story or a good plan, and they need just a component from us. We have a mix of projects. We have some kind of like the PCJ where we just stretch ourselves and kind of show what we can do and kind of take ownership and maybe co-partnering with, maybe be more of a co-partner with the collaborator. And others where we're just, we're happy to play a small but essential role, in helping them get through one step in the process. So I think, I think there are definitely avenues that require more work, more time, and it's a question of if the collaborator wants to allow us to be invested. If they really want it done right, then we have to sometimes draw the line and say, well, we need to plan this out. And so a lot of it, I think, is in terms of people and in terms of time, and maybe something in terms of the culture. And sometimes it's not always. People can understand maybe something tangible like an algorithm or a piece of software. But someone that does analysis, it's like people who write movie scripts. Some movies are terrible because they have a terrible script, but a Hollywood executive doesn't necessarily know a good script from a bad script or a good screenwriter from a bad screenwriter. Analysis can kind of work the same way, where I think maybe a culture where we kind of understand the value of analysis and giving people the freedom and the time to kind of do these deep dives. You have the data, but it's like, okay, people need time, and it's not just something kind of a purely technical role. I think having an idea of the higher questions, the MP questions, and trying to dig deep into those. Thanks.